Welcome to the lecture. In this lecture, we are going to discuss the various platinum drugs, levels and pharmacokinetics. Now, as we know that there is a various platinum drugs like cisplatin, oxaliplatin, nidoplatin, carboplatin. So these all are used as anti-cancer agent in clinic. Now, recently we are going to discuss the term that is a pharmacokinetics regarding with these three anti-cancer platinum drugs, how they can affect and how they can reach the cell and targeting cells. Let's see. Before that, we are going to discuss what is meant by the pharmacokinetics. As we know that the pharmacokinetics is the branch of to determine fate of substance. The fate of substance administered to living system and to determine and to discover the substance that it is administered up to the point at which it is completely eliminated from the body. Simply, we can say that the pharmacokinetics is nothing but how the organism affect on drug. So here, simply the pharmacokinetics is the how an organism affects on blood, affects on drug, and the pharmacodynamics is the study how drug affect on organism. So these are the pharmacokinetics and pharmacodynamics. So togetherly it can help in dose, affect dose, then how it will be affect on target cells. So now let's discuss the platinum drug levels regarding with this pharmacokinetics. As we know that the platinum analogous drugs are used for the anti-cancer as an anti-cancer agent. So it will generally affect so that the platinum drug analog is have been used as a drug in chemotherapy. Tumors. So, how they can affect the target cell? That is cisplatin, carboplatin, and oxaliplatin are first, second, and third generation platinum drug analog. These all three platinum analogs, platinum complexes. Platinum analogs means nothing but the cisplatin, carboplatin, and oxaliplatin are generally affect on DNA nucleus and on the other hand, the platinum-based anti-cancer drug interact with the target cells. So, for which the intracellular distribution of drug has already been investigated. The importance of cellular pharmacokinetics, subcellular distribution of platinum drugs. So after that, the platinum-based anti-cancer drug interact with cells for which the intracellular distribution of drug and around the cell target. Actually, platinum-based anti-cancer drugs like carboplatin, cisplatin, and oxaliplatin are generally target the cells, interact with the target localized cells, and for which the intracellular distribution of drug and concentration of around the cell target critical to efficacy. So now, what are the levels of these three platinum analog? That is the three platinum analogs. The platinum drug doses are 6.19 and 6.63 milligram per kg of that individuals. Blood sampling time were arranged as 0.08, 0.5, 1, 2, 4, 8, 12, 4, 48, 37 hours. So post dose for all three platinum based drug. So these are the hours by which we can give the doses of that type for carboplatin, oxaliplatin and cisplatin. Here 5, 6.19 and 6.36, 63 milligram per kg is that for 5 for cisplatin, carboplatin and for oxaliplatin. Each time point reach the blood samples were collected. Determination of or M.Sc.II. So these are all about the general determination of platinum drug levels and their pharmacokinetics. Now let's discuss the next. Now the next, the intact platinum based anti-cancer drug. Biotransfer into number of other platinum based but metabolites in vivo. Some of the platinum containing hydrolyzed product are effective as it can reach the target cell levels while others are not toxic. So these are all about the platinum anti-cancer drug levels and its pharmacokinetics. Now we will stop here. Thank you.